Okay, so this is epistasis in genetics from a computer science perspective. Epistasis is basically just the expression of alleles at one locus that masks or modifies the expression of other alleles at another locus. Basically, two genes are kind of fighting for the spotlight, and whichever one, quote unquote, wins is the one that we see in the phenotype. There are 6 different types of epistatic interactions between genes. There's different types: single recessive, single dominant, etc. And these phenotypic ratios over here on the side are pretty important because based on whatever our cross is, that's how we're going to be able to tell what type of interaction it is. And up here where there's no interaction, this 93 to 3 to 1 ratio, it's important as well because any deviation from that is what tells us that epistasis is occurring. We can visualize epistasis with these little maps right here. I like these a lot because I'm a very visual learner. And I'm also a computer programming minor and I like to code. So I thought these maps were very similar to how a computer scientist thinks while we're coding a program. So I decided to build a Java program that demonstrates duplicate recessive epistasis. And our pathway today is seashells that have phenotypes of purple or red shells. So duplicate recessive has a 9 to 7 phenotypic ratio. This is basically anything with a dominant A and dominant B allele automatically shows the dominant trait, which in this case is purple. It's really important that both dominant A and B are shown. It can't just be one or the other, it has to be both. So I'll show you the code I wrote. I like this program a lot just because I feel like it really replicates well how our body takes DNA and then kind of codes it into a physical manifestation of like a phenotype. And that's basically how coding works. You just type whatever you want and the computer spits back out what it gets. So up here, the user enters the seashell genotype, and whatever information you enter is going to go through these if statements, and depending on what it is, it's going to spit back out the proper phenotype. So let's test this out really quick. Our first cross is just a normal heterozygous parent cross where we're going to see the normal 9 to 7 ratio. So let's do big A, big A, big B, big B. Run the code, big A, big A, big B, big B, and we get phenotype of a purple shell. So for our second cross, this one's a little bit different. It's going to deviate a little bit from the normal 9 to 7 ratio because it's not a heterozygous cross. So this is just a big A, little B crossing with a little A, big B, little b. And we'll take little a, big B, little b for our test. So we'll just run the code again. We get little a, big B, little b, and we get a red shell phenotype, which is what we want. So again, I like this code a lot because I feel like it really represents the way our body kind of codes DNA. And this program really nicely mimics how these maps work whenever you're just working on an epistasis problem. So